morning, students. This is Murthy sir. Today we will learn about some practical observations. How to observe cells by using microscope in a laboratory. Let us observe the nucleus in human cheek cells with the help of a microscope. The materials required to do this experiment are microscope, a toothpick, glass slide, cover slip, watch glass, normal saline, needle, blotting paper, glycerin, and methylene blue. Methylene blue, it's a dye which is used for staining. Staining is a technique in which dyes like saffron and methylene blue are used to highlight particular regions of a cell or tissue. First, we have to take one toothpick and with the help of that toothpick, gently scrape the inner lining of the mouth and place this small piece of tissue in a watch glass containing normal saline. Next, we have to take a glass slide, and on that glass slide we have to put a drop of water, and in that the small piece of tissue which we collected from the mouth we have to keep in it, and in that with the help of a dropper we have to put 2 or 3 drops of glycerin so that the specimen which we want to observe will not dry up. On this specimen we have to keep one cover slip and we have to gently press on that, and we have to take care that any bubbles will not form. Now this glass slide we have to keep on the platform of the microscope. This cover slip which we kept on the specimen will not allow the specimen to come in contact with the lenses. Now we have to focus light onto the specimen with the help of a mirror which is present below, and we have to adjust the lenses to see clearly the specimen. Students, now you can observe cheek cells, and in these cells in the central region a spherical body is there. It is nucleus, the most important organelle in a cell. Now what you are observing is animal cell because we have taken cheek cells from our mouth. Now let us observe mitochondria in onion peel cells. The apparatus required for this experiment are microscope, glass slide, watch glass, cover slip, needle, glycerin, Xanus green bee dye, onion peel, and a blotting paper. The procedure of the experiment is just like the previous experiment where we observed nucleus in cheek cells. Now in this experiment we are observing plant cells, and in that you have to identify mitochondria. Actually, mitochondria are extremely small. They can be seen clearly in electron microscope. Their detailed structure can be studied with the help of electron microscope. Here what we are observing is small red colored dots in the cells. You can just identify their presence in the onion peel cells. Students, mitochondria are very small organelles present in cells for energy production. Here in onion peel cells, in all the cells, one clear spherical body is there. One big spherical body you can see in all the onion peel cells. That is nucleus. Nucleus is the most important organelle in a cell. It is called the control center of the cell. Nucleus controls all the life activities of the cell. So we are able to see clearly nucleus. But mitochondria, we are not able to see clearly. Here and there, red colored small dots are seen, that's all. But nucleus, we are able to see very clearly. A big spherical body is seen in all the onion peel cells. Students, now we will move on to the next practical observation, to observe cells in a leaf. Students, you know, leaf is a photosynthetic organ. You have to collect a thin leaf from the surroundings, any thin leaf, a grass blade or any leaf which is very thin, you have to collect from the surroundings. Leaf performs photosynthesis, it will do exchange of gases, it will do transpiration. It contains different types of tissues. A tissue is a group of cells which are similar in structure and function. Now let us see what is the apparatus required. A thin leaf, watch glass, glycerin, safranin is a dye, glass slide, cover slip, needle, dropper, microscope, blotting paper. These are the materials required for this experiment. So we will move on to the procedure. Procedure is actually like the first experiment which we have seen, that is observation of cheek cells, human cheek cells. So general procedure will be same, little differences will be there in the apparatus that we require for the experiment. So here with the help of a microscope we have to observe the different types of tissues in a leaf. Students, in this diagram you can see the different types of tissues in a leaf. It contains mesophyll cells, that is parenchymatous tissue, which contains chloroplasts. These chloroplasts are small organelles which contain chlorophyll pigment. In addition to that, you can see xylem and phloem tissues. Xylem transports water and mineral salts. Phloem tissue transports the food material that is prepared in the leaf. Leaf requires water and mineral salts, so that reaches the leaf through xylem tissue. In addition to that, you can see stomata, which are responsible for exchange of gases and transpiration. So upper portion of the leaf contains more number of chloroplasts, so that the leaf is exposed to photosynthesis. Students, now we will move on to the next practical observation, to observe cells in a dicot stem. You know, stem is an important organ in a plant body. It is a vegetative organ, just like root and leaf. The stem contains different types of tissues. To observe that, the apparatus required are a thin slice of dicot stem, watch glass, blade, needle, glass slide, glycerin, cover slip, safranin or methylene blue dyes, dropper, microscope. So with the help of this apparatus, we have to observe the different types of cells and tissues in a stem. So here we are preferring dicot stem. For example, sunflower stem, if you observe, it is a dicot stem. So we have to use microscope here, compound microscope. It is also called light microscope. In this microscope, we have to focus light onto the specimen. So in this diagram, you can see the different types of tissues in a dicot stem. Students, here we will observe different types of tissues in dicot stem. Epidermal tissue, which is for protection, collenchyma, parenchyma, sclerenchyma, chlorenchyma. These are all different types of tissues that are seen in a stem. In addition to that, vascular tissue will be there, just like a leaf. Here also xylem and phloem tissue will be there. The xylem transports water and mineral salts, phloem transports food material. A stem, mainly it supports the transport of water, mineral salts, and food material. In addition to that, stem plays an important role in the development of lateral branches. A stem supports the growth of lateral branches. So concerning that, different types of tissues like parenchyma, collenchyma, sclerenchyma, chlorenchyma, all these tissues will be seen here. Vascular tissue also there, just like leaf, xylem and phloem tissue. Xylem transports water and mineral salts, and phloem transports food material. So here, tissues, you can see a tissue is a group of cells which are similar in structure and function. So next to cells, tissues will be there. This leaf, stem, root, flower, they're all organs. A group of tissues will form an organ, and different organs will make up the plant body. Root, stem, leaf, and flower, together they make up the plant body. So here you can see the definition of tissue. A tissue is a group of cells which are similar in structure and function. Different types of tissues together will make up organs.